In this lecture, we're going to briefly review vesicular transport. Remember, last lecture we discussed import of proteins into the nucleus and across membranes such as the mitochondria or the ER. Proteins that are made in the ER then will need to be potentially delivered to other locations. Movement of proteins to another location, such as from the ER to the Golgi or the Golgi to the membrane, requires vesicle formation. In addition, proteins that actually move in from the cell surface into cells and other types of substances will also be moved in, which is called endocytosis, also by vesicular transport. So when we think about different movement of vesicles in cells, two different processes involving vesicle formation and the directional movement of vesicles is endocytosis and exocytosis. So if you look here to the right, this diagram is showing you endocytosis, the inward formation of vesicles. This is used in different ways to ultimately bring in materials, either specifically due to binding of certain receptors or more non-specific movement of vesicles such as pinocytosis. And also endocytosis, another form, is phagocytosis, which is accomplished by immune cells in our body. We also have exocytosis, the exiting of vesicles, exocytosis, the exiting of vesicles, such as at the trans-Golgi, release of membrane proteins, other types of substances to the cell surface. And we also have a secretory pathway of exocytosis involving secretion of particular substances by specific secreting cells. If you'll notice also, we have vesicles that are forming obviously at the surface of our ER to allow for movement of proteins from the ER into the Golgi, and also there's vesicles that move backwards as well to move proteins back to the ER, etc. So when we think about vesicle formation of these small transportable membrane-bound compartments, the formation of these vesicles has been shown to due to binding of proteins that are called coat proteins. Coat proteins are ultimately have two distinct functions, one of which is binding of specific proteins to form these outer coats, help induce curvature of the membrane to form the budded vesicle. And another important role is they're involved also in selection of specific cargo into these vesicles, these form-budded vesicles. When they look at electron micrograph images of vesicles, they see this fuzzy material that is on the surface of the membrane. This fuzzy material has now been found to be different types of protein coats or coat proteins. And there's different classes of coats, proteins that have been identified. This particular diagram is showing one particular class of coat proteins, which are called COP2 coat proteins, and their assembly process, which we're going to talk about a little bit later. Now, 3 known types of coated vesicles have been well studied. There may potentially be others, but these are the 3 that are most well studied. COP2 coated vesicles, which are important for movement from the ER to the ER Golgi intermediate complex and to the Golgi. So movement in this particular direction involves COP2 protein coat assembly. COP1 coated vesicles are thought to be important for back traffic, so backwards from the Golgi to the ER and essentially in that particular direction. So COP1 appears to move retrograde, so move an opposite direction. Clathrin-coated vesicles have also been well studied, and this particular coated vesicle is thought to be important for movement of material from the trans-Golgi network to lysosomes, endosomes, and also been found to be important in movement to the plant vacuoles, as well as its Clathrin-coated vesicles also form at the surface or the membrane surface during endocytosis. Now, just to give you an idea of how these coat proteins work to force budding of a vesicle, so formation, membrane, force the membrane to form into this curved, smaller circular sphere of membrane. one particular coat protein is the COP2 coated vesicles. And remember, they're moving material essentially from the ER to the ER intermediate Golgi complex, and also to the cis Golgi. One important player in this particular assembly process is a G protein called SAR1. SAR1 will exchange GDP for GTP, which is stimulated by a guanine exchange factor, which is shown here in the membrane. When it's bound to GTP, it has a finger-like helix that forms that inserts into the membrane. This insertion into the membrane causes slight curvature of the membrane. This then leads to binding of COP2 coat proteins, and particularly a dimer of what's called SEC23 and SEC24 will interact with SAR1 to start to form additional curvature to the membrane. So binding to SAR1, GTP, will start to curve the membrane even more. These coat proteins, SEC23 and SEC24, also have an important role because they are also going to help select for specific cargo through the C-terminal tail end or domain of the particular cargo receptors. SEC24, there's different versions of these SEC23, SEC24 proteins to allow for specific selection of certain cargo. Then, following the SEC23 and SEC24 assembly and selection of specific cargo into a vesicle, there's also binding of additional coat proteins, which are called SEC13 and SEC31. These will assemble as an outer coating to help form additional curvature of the membrane to form the vesicle. So essentially these coat proteins are forcing curvature of the membrane, but also allowing for selected cargo placement into these vesicles. Now, ultimately, the SEC13 and SEC31 will help form the full curvature. The vesicle will become released, and then disassembly of the coat then occurs due to hydrolysis of GTP on the SAR1 protein. Hydrolysis of GTP to GDP of SAR1 will lead to disassembly of the protein coat, which happens shortly after release of the vesicle. This particular model is showing you what is thought to be the SEC1331 cage of the COP1 coat after assembly onto the final vesicle. In this particular case, the surface of the vesicle is 40 nanometers. It forms this four-legged structure due to interaction of these SEC13, SEC31 subunits. So we get this specific cage-like structure that's ultimately going to help form that vesicle shape, and this of course will disassemble shortly after release of the vesicle. Another well-studied protein coat is the COP1 protein-coated vesicles. COP1 coated vesicles actually work in the opposite direction as COP2 coated vesicles, and this type of protein coat will assemble onto material that needs to go backwards from the Golgi and specific stacks of the Golgi back towards the ER. This is important for retention or bringing back proteins that need to stay in the ER. So it helps return proteins back to the ER, whether they're proteins that are involved in docking or other types of proteins that accidentally got placed into a vesicle that moved to the ER. Ultimately, these particular types of proteins have special tags that allow them to bind to certain receptors, so they get packaged into these COP1 coated vesicles and returned to their proper location. COP1 protein coat assembly is very similar to COP2. There's specific subunits that help form the outer coating, and there's also a special GTP protein that's called ARF1 that's like SAR1 in COP2. ARF1 is similar in the fact that when it binds GTP, it will also insert into the membrane, starting the curvature process, and then also allow for recruitment of the specific COP1 coat binding proteins to force the additional curvature plus selection of cargo. In addition to the COP2 and COP1 protein coats, there's also what are called Clathrin-coated vesicles that have also been well studied. Many of the Clathrin-coated vesicles, the analysis of these vesicles have been shown looking at endocytosis of particular material. Clathrin-coated vesicles are not only involved in movement of vesicles or material into the cell, but are also important on the trans-Golgi and formation of vesicles at the trans surface with movement to the lysosomes and endosomes, as well as you saw earlier for plant vacuoles. So this particular image is showing you increase in time in regards to formation of Clathrin-coated vesicles. This happens to be in uptake of specific yolk lipoproteins by hen oocytes, and these were electron micrograph images showing you at specific time points and further in that process of vesicle formation of Clathrin-coated pits. Clathrin proteins, when they assemble to generate the vesicle formation, they have a unique basket-like array, and we're going to look at a predictive model of those proteins in a second. Now, Clathrin-coated vesicles to ultimately form involve specific coat proteins that force the shape, just as we saw with the COP2 and COP1 protein coats. They also have important proteins that are going to be recruiting specific cargo, like we saw with the SEC23 and SEC24 in coat 2 protein assembly. So specific adaptant proteins are going to be important in binding to the coat proteins as well as selection of a specific material, also through the C-terminal domain of those particular receptors. So we see the green protein coat assembly as well as the involvement of specific adaptant proteins, which you see binding to specific cargo. So there's going to be a selection process as well that allows for a certain selection of material into these vesicles, and the coat proteins are going to form that spherical shape. Now, something different that has been discovered with Clathrin vesicle formation is the role of an important G protein called dynamin. And dynamin is going to be responsible for release of these Clathrin-coated vesicles. So we'll talk a little bit more about that in one of the upcoming slides. Also, after the protein coat has formed for Clathrin-coated vesicles, these coat proteins are removed prior to delivery or docking to their target membrane. So Clathrin-coated vesicles, the protein coat is assembled through these triskelion formation of protein assemblies. And these triskelion units are comprised of 3 heavy and 3 light chain subunits. They form this triskelion, this tri-complex protein structure, which you can see here in the electron micrograph. These will assemble to generate a basket-like array that will force the shape of that vesicle surface. Keep in mind also, like I described before, special adapter proteins, for example, AP2, and there's been several other types of adapters that have been discovered as well, are important for selection of particular cargo. In this particular scenario you're seeing here, adapter protein AP2 selecting a particular receptor within the membrane, which allows for particular selection of not only transmembrane proteins, which could be potentially the receptor, but also cargo that's bound to that particular receptor protein. And you can see the Clathrin-coated proteins will bind to these adaptants to force the curvature. Binding of these adaptants requires special inositol phospholipid molecules, some of which your textbook talks about a little bit, and the literature also talks about even more so. Now remember, I told you release of these vesicles appear to be different than the COP2 and COP1 protein-coated assembly vesicles. It involves a special complex of what are called dynamin proteins. Dynamin proteins are G proteins. They start to assemble as the vesicle starts to be completed in its budding process, and they assemble by linking together. So multiple dynamin subunits link together into a chain, And this chain will essentially start to wrap around the membrane that remains, that allows for attachment of that vesicle still to the surface of something like the trans Golgi, for example. The assembly of these dynamin subunits force a ring structure around the membrane. Hydrolysis of GTP to GDP causes a change in that coiled ring structure to where it tightens around that membrane, forcing release of the vesicle, which is shown here. In fact, there's been a few different experimental studies working with non-hydrolyzable GTP analogs that allow for dynamin to bind GTP but not hydrolyze GTP, and there's loss of that change in that conformational, that spiraling of that ring structure, which leads to severing of the membrane. And what happens is the dynamin subunits keep adding and coiling, and you get this long extension of the membrane with the budded vesicle, but you do not get release, which is shown here in this particular diagram. So ultimately, dynamin and hydrolysis of GTP leads to a conformational state change of this ring structure and allowing for severing of the membrane. Another term for release is called fission, so you might run across this in different papers as well. Now, different protein coats are used to assemble the membrane as well as select specific cargo at various locations. But other classes of proteins are important in ultimately helping dock the vesicle so it can fuse with the target membrane, such as movement of vesicles from the ER to the Golgi, or from the trans Golgi to the endosome, etc., so that that material can then be incorporated into the next compartment, and potentially those proteins may be further modified and then moved on further later by, once again, vesicle formation and release. These proteins that are important for docking are called snare proteins, and they're what are called V snares and T snares. And these proteins are also inserted into the vesicle when the vesicle is being formed, which is also part of some of the selection process for that formation of that vesicle. V snares are on vesicles, which is where we get the V. T snares being target membrane, which is where we get the T. There's been different V snares and T snares discovered that are important for certain types of membranes and incorporated into certain types of vesicles, which you can see these V snares and T snares at a particular target membrane. In addition to snares, these proteins that are important for docking and fusion of these vesicle membrane to the target membrane, there's other classes of proteins that appear to be similar as well, important as well in this docking process. Many of which are what are called Rab proteins, which are also G proteins, and also what are called tethering proteins. Rab proteins mediate binding and assembly of specific tethering proteins. And you can see there's different types of tethering proteins that have been discovered. Some are more filamentous that link together in chains that literally hold or help hold the vesicle in place so ultimately it can move forward and allow for interaction between the snares to cause fusion. So tethering proteins could stretch a little ways from the membrane to really help bring in that vesicle. There's several different types of snares that have been found or discovered and that are involved in the fusion process. And interaction between these two, V snares and T snares, will help drive fusion once that vesicle is near that target membrane. This particular model is describing how snares might mediate fusion. And it is an ongoing model, so potentially it could change. But ultimately, the vesicle starts to interact with the target membrane through interaction between these snare proteins, which involves formation of what's called a four-stranded protein bundle, which involves interactions between helices, which you can see here. That forces the vesicle to come in a very close location or close proximity to that target membrane, which will then help mediate fusion. One possible model shows that ultimately first in that fusion process is there's a formation of a pore, which you can see here, to allow for fusion of that target membrane. And you'll notice that V snares and T snares end up together in that target membrane. And ultimately those V snares will then have to be moved back to the original membrane so that they can ultimately be incorporated into vesicles again. Now keep in mind, movement of these vesicles once they've formed oftentimes is mediated through motor proteins, and essentially different types of microtubule motor proteins. Dynein motor proteins are thought to move material in what's called retrograde, such as movement of material from the Golgi back to the ER for retention of particular proteins that are needed to be retained in the ER, or movement of specific receptors, or also V snares back to their proper location. Kinesins move towards what's called the plus end of microtubules, which essentially move in what's called the anterograde movement, which is moving towards from the ER to the Golgi, the trans Golgi to the endosome, etc. So that would be the directional motor protein that moves it in that particular direction. So vesicular movement to their proper target membranes is thought to be mediated due to binding to specific motor proteins through specialized proteins, which you can see here. And ultimately, different classes of motor proteins can move in different directions or move these vesicles in different directions, as you can see from this particular diagram. So this completes our shorter lecture on vesicular transport, just giving a summary on how vesicles are formed, and ultimately the different types of protein coats and vesicle formation, as well as delivery of these vesicles to target membrane through tethering proteins, as well as docking using snare proteins, which helps mediate fusion, and ultimately the role of motor proteins in moving these materials to their proper destinations.